Now, some more questions. So, mention two limitations of inferred functional profiling using whole genome sequencing data. Inefficient in two limitations of inferred functional profiling in whole genome sequencing. So, the first is inefficient in low microbiome biomass environments due to host contamination, and also inefficient recovery of rare species. Now, the database used for out reference in Picrosty for functional profiling is Green Genes, and for the other one is IMG image. MAGs are generated by assembling metagenomic reads, metagenome assembled genomes into context, and then binning them into individual genomes. Assembling them into context and binning these contexts into individual genomes. Botai 2 uses hidden Markov model to identify motifs both for distant homologs as well as for accurate annotation of functional domains. Right. Moving on, Botai 2 uses hidden Markov model and a graph formalism in which paths represent contiguous genomic sequences is constructed from overlapping k-mers during assembly, known as De Bruijn graph. So that's it. Moving on to another set. Which of the following statements is false when using longer k-mer sizes of the De Bruijn graph? Which is false. So basically, it results in more loops with longer k-mer sizes. That is false. Longer k-mer sizes of De Bruijn graph is better at handling repeats. That is true. It is more likely to contain errors leading to loss of sensitivity is also true, especially worse for rare genomes. That is true. This is the only statement that is false. It does not result in more loops. Rather, smaller k-mer sizes of the De Bruijn graph would lead to more loops than the longer k-mer sizes. That's why this is the answer. Now, which algorithm does MetaVelvet employ for metagenomic assembly? The answer is De Bruijn graph. Which of the following best describes the methodology of PanFAN? That is, it is read-based and gene family detection based. That is, whether we have absence or presence of the gene family. What is done to the edge with the highest betweenness in the edge betweenness algorithm for module detection in a network? It is removed, potentially breaking the network into subcommunities. Right, so that's the answer. Now, a species by default secretes a specific metabolite that reduces the growth of some organisms in the community. The secretion is non-specific and not directed at any specific species. The interaction is amenalism. What is the application of walk trap approach in network analysis? That is subcommunity detection. Which is the test used for continuous metadata variables when the distribution is unknown and data is sparse? So the answer is Kendall-Tau. Spearman row is basically when the distribution is unknown and the data is non-sparse. So that is for Spearman when data is non-sparse. When data is sparse, we have Kendall-Tau. And when the distribution is known and normal, then it is Pearson. Which of the following tool utilizes training model derived from datasets from a specific environment to infer functional profiles from new taxonomic profiles? That is MELANPAN. Now, which is indicative of higher growth rate of given species, high peak to trough ratio of mapped reads. Now, the assembly of metagenome in assembled genomes from metagenomic reads datasets for a given species is dependent on two factors. First is abundance, and second is genome length. The assembly of metagenome assembled genomes is dependent on abundance and genome length. Then Concoct utilizes a Gaussian mixture model to identify the ideal number of clusters or bins. Moving on, what does the output of the number of polymorphic sites or non-polymorphic sites of a given species convey in strain plan? That is alpha diversity of strains of that species in the microbiome. Moving on, the process where reads from all samples are merged and then assembled is called co-assembly. Right. PrEP is a module in R that can be used for building networks using the reboot approach. That is renormalization, permutation, bootstrapping. Number of shortest paths that pass through a particular node is a measure of the betweenness centrality for that node. Number of shortest paths passing through that particular node. Now, difference between multi and co-assembly. So multi-assembly is basically assembling each sample independently and then forming a multi-assembly from the individual assemblies. And co-assembly is actually merging reads from all samples and then assembling. So we merge reads from all samples here in co-assembly, and in multi-assembly we assemble each sample independently. Now name the database used for OTU references in Picrosty for functional profiling, that is Green Genes database. Now competition is the name of interaction association where both community members suffer loss. Name two applications of generating metagenomic assembled genomes. So that is functional and metabolic capabilities of individual community members, identifying novel lineages of species and profiling strain specific variations. So that's it about the video.